Okay, so this is chapter 3, part 4, and now we're going to be talking about proteins. So here we are, it's another macromolecule. So this is our third biological organic compound. And proteins are incredibly important. Not to say that lipids or carbs aren't either, but proteins are very important because they really have so many different roles, so many roles inside of organisms. Many of them are enzymes. And now, if you're not familiar with what enzymes are necessary for any kind of chemical reaction to happen in our body. And there's millions and gazillions of chemical reactions happening in our body. And these chemical reactions would literally not happen if enzymes weren't present, right? So even just like breaking down something, so lactose, remember lactose, if we want to break it down into its individual monosaccharides, that's a chemical reaction, and we need an enzyme in order to carry out that chemical reaction. So enzymes are basically predominantly made out of proteins. A lot of our hormones are also proteins as well. We have our growth hormone. Insulin is also a protein, right? So it regulates the blood sugar levels in you. There's lots of ones that are involved, in transport. So they move things around, they move other molecules. Like hemoglobin, for example, is found in your blood, and it moves oxygen around our body. So without that, we would be in a very bad situation. It helps with contractile movement as well. So myosin and actin are two kinds of proteins that help your muscles actually contract. Protection, so your immune system, you have antibodies. These are made out of proteins, right? So they defend against bad things. They defend against invaders. They're also important for structure. So anything like your skin has keratin on it, that's a protein. Your hair, right? Your collagen is also a protein. So in your cartilage, etc. Storage as well. It can help store nutrients. So you can think about the egg yolk, and sorry, not the egg yolk, the albumin, so the white part of the egg, right? That's actually technically the egg yolk would turn into the little chick, right? And the egg yolk, the white around the egg yolk is technically a protein that provides nutrition for the growing chicken, or the growing chick. They can also be toxins, so lots of different kinds of animals and various organisms secrete certain proteins that act as toxins, and they're very bad for us. And then the example they give here is bacterial diphtheria toxin. That's actually, so it's given by a specific bacteria called Corynebacterium diphtheriae. This bacteria causes basically diphtheria. If you have it secretes a toxin that causes you to form a weird, like a false membrane across your throat, and it stops you from breathing. So really bad toxin. You basically die of asphyxiation. Not good. And then in addition, proteins can also work with communication or cell signaling, right? So they communicate. They're the actual communicators between different cells and different tissues. So proteins have a variety of roles in an organism, and they're incredibly important. Proteins also have their own specific monomers, right? The monomers that make up a protein are known as amino acids. So we have 20 different kinds of amino acids. And if we see here, these amino acids, there we go. They're all very similar looking. The only thing that they have that's different is the highlighted areas around them, on them. But everything else about an amino acid is similar. So all these monomers are just different kinds of beads. These amino acids are the building blocks, are the beads for the protein. And each one gives a specific property to the protein, right? So the amino acids come together, and they come together through a specific kind of covalent bond called a peptide bond. We'll look into it in just a second. When we have a chain of amino acids, so they're all bound together, we form a polypeptide chain. Okay, so that would be all of your amino acids in a row. This is a polypeptide chain. Once this polypeptide chain then folds on itself, and it forms a specific three-dimensional structure. So here I have a weird little U that I've made, okay, a specific three-dimensional structure. That is when we have a functioning protein. A protein cannot function unless it has a specific structure to it, because usually, again, the structure is going to dictate what it binds to, right? So this can bind to this. If the structure was like this, it wouldn't bind to whatever it needs to bind to, right? But here, I don't know if I'm going to be able to do that again. With this specific structure, we can now bind, like this, it can actually bind to something and, you know, engulf it or whatever it has to do. Okay, so the sequence of amino acids just as is, when they're all linked together, is known as a polypeptide chain. And then once this chain actually folds in on itself and has a proper 3D configuration, that's when we get an active functioning protein. Okay, and this shape is really important. Shape is critical to the function of all proteins. It's basically like a hand in glove fit, right? Your hand would not be able to fit inside a glove if the glove wasn't shaped like your hand. It's the same thing with proteins. They wouldn't be able to act on other things and do carry out their roles if they weren't shaped to basically kind of go around, like, you know, like to fit around their target. So protein shape is very important. And like I said, the amino acids are the building blocks or the monomers or the subunits of the protein. So they all have to be added together. So let's look at these monomers in a little bit more detail. So there's 20 different amino acids, right? We saw them here. No, you do not need to memorize any of them, okay? You don't need to memorize them. What you do need to know is the things that they have in common. So if you see here, each one has a carbon. Each one has what we call an amino group, this NH3 right here. Each one has a carboxyl group, the COO right here. And then each one has a hydrogen. And then the only thing that's different between each amino acid, so here we have glycine, alanine, valine, leucine, etc. The only thing that's different is this bottom side chain right here. Glycine only has a hydrogen. Alanine has a CH3. Valine has this whole weird nonsense thing right here. Tryptophan, okay, it has this weird aromatic configuration. So the only thing that's different between these 20 amino acids are these side chains. And we also call them, we basically denote them by the letter R just because they're random, right? They're random for each amino acid. Okay, so it's you can think of the R as the rest of the molecule. So we have 20 different kinds of R's, so 20 different amino acids. So this, you can see that they all have an amino group, this part right here, a carboxyl group, this part right here, a hydrogen, a carbon in the middle, and then this side chain right here, okay? So each type of amino acid has a unique side chain or R group, which gives the amino acid its special chemical properties. So this means we have some amino acids that are non-polar, so they don't like water, some that are polar, so they like water, some that are acidic, some that are basic, some that are negatively charged, and some that are positively charged. So each one, depending on the actual side chain that it has, right, has a specific property. And this is really important because the sequence of these amino acids is going to dictate how our protein folds, how our polypeptide chain folds. Okay, so for example, I'm just going to switch over my camera real quick. For example, if I have a sequence of amino acids, so amino acid 1 here, okay, amino acid 1, amino acid AA2, amino acid AA3, okay, amino acid AA4. I may even repeat amino acid 1 here. Oops. Sorry. Who knows? It's just completely random. And here I have amino acid 5, amino acid 6. Okay, so these amino acids are all linked together. The link that holds them together is a covalent bond that is specifically, in the case of proteins, it's specifically called a peptide bond. And so this whole chain of amino acids, say this chain of 7 amino acids, is known as a polypeptide chain. Okay, but this is still not a protein. In order for this to become a protein, it needs to fold up properly. And the way it folds is all dependent on its actual amino acid sequence. So let's say, for example, this amino acid right here is positive. It has a positive charge. Okay, and this one as well has a positive charge. This one, let's pretend, is neutral. This one is something else, whatever. This may be positive as well because I repeated it. Okay. And amino acid 5, let's pretend, is negative. Amino acid 6, let's also pretend it's negative. What do you think is going to happen, right? Probably what's going to happen is that this positive side right here is going to be attracted physically, right, like a magnet, to this negative part right here. So I think, and this is exactly what's going to happen, This particular seven amino acid long polypeptide chain is going to fold up in such a way that the positive amino acids, amino acid 1 and 2, are going to be bound, like are going to come together to the negative amino acids, amino acid 5 and 6. And the other ones are going to basically loop out like so. Do you see that? I still have 7. 1, 2, 3, 4, 5, 6. Sorry, 1, 2, 3, 4, 5, 6. I wrote too many. I drew too many. I'm sorry. Here you go. I'm just going to make this one a little bit bigger. There you go. So 5, 6, 7 amino acids. All right. But because this side is positive and this side is negative, we now have a specific configuration. Now it's folded on itself. So my overall structure of the protein looks like this. It looks like a weird little jelly bean. I don't know what that looks like. It's like a Christmas stocking. I don't know. Okay. So this is a specific 3D structure of my protein. And so now I have a protein that is functional. Okay. This protein can actually be biologically active and functional because it has a 3D shape. Oops, shape, there we go. Okay, so this is why it's important to know that each amino acid has a specific property to it, because these properties dictate how the protein is going to fold. So when two amino acids are actually joined together, remember they're monomers, and monomers, in order to come together, they have to go through a dehydration reaction. So the amino acids, again, hydroxyl group is removed from one monomer, from one amino acid. The hydrogen group is removed from the other amino acid. So water comes out, and then these two come and join together. And this right here is known as a peptide bond, okay, between the carbon and the carbon of one amino acid and the nitrogen of another amino acid. We have the peptide bond. Okay, so so because of the R group, remember, is a side chain that may be non-polar, so not charged, and they, hydrophobic, uncharged but polar, positively charged, negatively charged, acidic or basic, lots of different varieties. Proteins use basically these 20 different letters or amino acids to create polypeptides, hundreds and thousands of amino acids in length. The amino acid sequence of each polypeptide determines the three-dimensional structure of the protein. A protein's 3D structure enables a molecule to carry out its specific function. And then nearly all proteins work by recognizing and binding to some other molecule. So this is why the three-dimensional structure of a protein is so important. Okay, so here I'm just showing you how we have the addition of this amino acid, methionine, plus alanine, plus leucine. To add it to now tryptophan, we have to do a dehydration reaction. Again, I don't need you guys to remember, I know the amino acids because I've studied this all my life, essentially. You guys don't need to know the actual types of amino acids. Just know what amino acids are in general, right? And how do they come together? And when they come together, what molecule do they form? And they form a polypeptide chain, and then this polypeptide chain needs to be properly folded to make a protein. So definitely look at this animation. I'm not going to actually play it right now. So there's animations. There's actually 2 animations in your PowerPoint waiting for you. Okay, so this is an example of a protein. I mentioned this one before. Hemoglobin is the one that we find in our red blood cells that actually transports the oxygen for us around our body. And if you see, this is what hemoglobin looks like. And it's really great because basically our oxygen can get nestled right in the middle of this little nest that hemoglobin makes. And this is how oxygen gets transported. And if you actually, this is another thing that you should look, you should be able to notice, is that the protein, the hemoglobin protein, is made out of 4 different polypeptide chains. It's made out of this yellow one, this orange one, this blue one, and this green one. So there's actually 4 different polypeptide chains that come together to form a protein. And this is something that happens. Not all proteins are made out of one single polypeptide chain. Most proteins are actually more than 1 polypeptide chain. So there's like at least 2 polypeptide chains. So like collagen, for example, is 3 polypeptide chains. So hemoglobin, we have 2 alpha globin polypeptide chains and 2 beta globin polypeptide chains. And they come together to form this really cool little nest, right? So the 3D structure is really important. And then in this 3D structure, this is how hemoglobin can actually bind to oxygen. Okay, so another reason why I brought up hemoglobin is because it kind of shows you how important it is to, how important the actual specific sequence of a protein is. and how one small little change, one small change in amino acid, like a one amino acid change, can really distort the entire protein. So maybe you guys have heard of a genetic disease called sickle cell anemia. Okay, so people with sickle cell anemia have a mutation, and this mutation carries into their hemoglobin protein, so that the hemoglobin protein doesn't form, its structure is a bit wonky, and it doesn't basically transport oxygen very well. So before I get there, let's just go over what normal hemoglobin looks like. So a person that doesn't suffer from this disease, they have normal hemoglobin, which looks like this, that really weird, messy, like nest, right, where oxygen kind of goes in. And with normal hemoglobin, it works perfectly fine. It binds to oxygen, and you have it in our red blood cells that have hemoglobin inside of them. Our red blood cells look like that really tasty, that sounds really weird, but to me, they look like jelly donuts. Okay, so that really cool structure, that jelly donut structure that we know so well with red blood cells, right? Now, if we go and look at the mutated version of hemoglobin, which is what people with sickle cell anemia have, they literally have, you can't really tell in this, there we go. They literally have the only mutation that they have compared to, like, you see, this is the amino acid sequence. This is a polypeptide chain of the normal hemoglobin, okay? Between this sequence and this one, if you can spot the difference, the only difference is this one right here, okay? There's only the 6th amino acid, instead of being a glutamate, is a valine. Again, you don't need to remember the specific amino acids, but just know that there's one amino acid that is different in the entire chain, and there's about 150 amino acids in this whole chain, right? There's only one small amino acid that is different. This one single amino acid difference distorts our hemoglobin. Can you see how the structure of our hemoglobin is a lot more like circular and less like a nest than this one up here? If you can't see that, you can definitely see that they are different. The shapes are different from each other. Because of this difference in shape, hemoglobin doesn't act properly anymore. And a lot of people are basically, anyone with sickle cell disease is anemic, meaning they don't have oxygen, can get to their organs quite as well, right? And in addition, because of this weird structure of hemoglobin, this mutated hemoglobin structure, our red blood cells, people with sickle cell disease, their red blood cells look like a weird little sickle shape, right? And this makes the red blood cell a lot more fragile. So the red blood cells literally pop way more often. They just pop. They just lose red blood cells, and it causes a lot of, they can clog up. If you see here, yeah, these sickled red blood cells they're a lot broader and just like bulkier than our normal red blood cells, and so they can clog up arteries really quickly. So people that have sickle cell anemia usually suffer from a heart attack around age 30, okay, age 30 like to 35, so very early in their lifetime. And this is all because of one single mutation, one little mutation, one single wrong amino acid. So again, this basically underlines how important it is for the sequence of the sequence of amino acids to be in a specific arrangement, because that arrangement is going to dictate how the protein folds. And if the protein folds incorrectly, that protein isn't going to work anymore. We need a correct three-dimensional configuration for a protein to work. Another thing to think about to basically underline the importance of proteins and their structure is actually with prion disease. You may have heard of mad cow disease, right? Mad cow disease, we weren't allowed to eat cow because we would take up a protein that would make our own proteins go crazy. And in the end, it would essentially basically eat away at our brain. It's actually really nasty. This all happened because proteins were misfolded. So this is a disease of proteins being misfolded, and just because they're misfolded, they aggregated with each other. They basically clustered up, and they cause a lot of problems, like prion disease and Creutzfeldt-Jacobs disease and mad cow, right? So protein folding is very important. So we do have to be wary of our proteins not folding correctly, right? And there are certain circumstances that can basically denature our proteins. The word denature means they damage and they take away our protein from its original natural state. Denatures, takes it away from their natural state. So that protein three-dimensional configuration can come undone and it can become damaged. This is because their shape is very sensitive to the environment. So any change in pH, right? Any change in temperature that they don't like, or some other kind of factor like a chemical or a toxin, this can disrupt the three-dimensional configuration of the protein. It can break all sorts of bonds. It can break those like the 3D structure, and the protein unfolds. And when the protein unfolds, it denatures. What do you think? Do you think that when the protein unfolds and it denatures, do you think that the protein is functional? I hope you said no, right? The protein, as soon as it unfolds and it gets denatured, it no longer works. And so the few things cause it to be denatured, like changes in pH, temperature, and then other things. A really good example is an egg, right? So I mentioned how the white part of an egg, that slimy mucousy part of an egg, is a protein. Now, you've seen what an egg at room temperature looks like. That white part is just a mucus, nasty little thing. When you heat it up, like when you fry an egg, that white part, or boil it, that white part becomes actually like it solidifies and it becomes white. That's because that change, that actual physical change that you saw in color and in texture, occurs because our protein, the albumin egg protein that we had in the mucus, was denatured with the heat. Okay, and then yeah, so this is also a reason why when we have fevers, it can get really bad. Like if we have too high of a fever, we go to the hospital, because too high of a fever, we increase our temperature too much, and this could affect all of our enzymes and all of our proteins in our body and could cause our body to shut down. Not a good thing, right? Changes in pH as well. Proteins usually function at a specific kind of pH, right? So the proteins in your stomach, they function at a very acidic pH because your stomach is acidic. Proteins, some usually like in your blood or some other places, they function at a neutral pH. If you take that protein away from its optimal environment and put them in so its optimal pH, and put it in a pH that it does not like, it's going to denature, it's going to unfold. So proteins, that's basically the end of proteins. Okay, I'm not going to draw another little concept map. I'm going to make you draw the concept map for proteins and maybe even show it to me at our Zoom meeting. Remind me, because I'm going to forget. But yeah, draw a little concept map. I challenge you to draw a little concept map about proteins. Go over things like what are its building blocks, what bond binds the actual building blocks together, what does it form, what chain does it form, what then has to happen in order for the protein to become functional, so it has to fold, right? And then what could cause this folding to go wrong, so like mutations, denaturation, explain what those are. So I definitely think that you guys should try this, take up this challenge and see, what kind of concept map you make, right? And you may even be awarded points for it depending on how good it is. So definitely take your time, make it colorful as well, right? Different colors pop up, and this is a great way to study again. Like this is a very simple thing to do, but you will be surprised at how efficient it is. So I'll see you guys in the next video, and the next video will be the last part on chapter 3.